quite sure why we did bitterness, but I feel like I don't know, it's maybe easier to measure. And we also like beer and IBUs like go hand in hand. That's how we measure the flavor, I guess, through IBUs. I'm not 100% sure.
Sounds great. Thank you.
Yeah, sorry.
Oh, it's still a good question.
I feel good. I feel like that was great.
Thanks.
Did the local brewery have more of a it looks like there's some variation, but you said earlier that there wasn't that much based on their different yeast strains within it. Is that what I took away from your talk?
Say that one more time. Do
some of the smaller breweries like this, local brewery here, it looks like from some of your data that in terms of the international bitter units that it does have some variation within it. And do they think that's more of a yeast derivative or an ingredient change, I guess I should say?
Maybe both. I'm not sure I'm understanding the question, but
I guess so. I was just looking at your graphs here. The blue
right.
lines and the orange lines for
right.
those are different days. It looks like on different, the 16th of March and the 30th of March, but those have at least some of those profiles there. Are those different batches? They have slightly different IBU units. Could that be due to the ingredients that the brewers put in, or due to the yeast making those IBUs?
I feel like it's more the ingredients, because Megan was talking about like the hops that they put into it.
Okay.
And so the IBUs kind of, and I think at the last 5 here, she was saying that there wasn't too much of a variation. The first ones, I'm not sure what she did with those. I think those were just different samples from like the store.
Okay.
Just regular Coors Light or whatever. Yeah.
Okay. Just curious. And then so the goal here is also to on your gel electrophoresis there is also to see if you can monitor different like ratios of the yeast strains.
Not necessarily. We're basically just trying to make sure that there is no contamination.
Okay.
So in the gel, yeah.
Okay. So you're developing an assay so you can test that though.
Yeah, we could test that. We're trying to see if there is contamination between these beers, because we don't want that.
Okay. Yeah, that's okay. I got that now. Thank you.
Yeah.
Okay. Excellent presentation. So Alexis, thank you very much. Please, everyone give a round of applause.
Thank you.
Even though it's digital.
Yeah.
So thank you very much. Our next presenter is also from UCCS. Her name is Annalise Calzadilla, I believe. Is that correct?
Super close. Calzadilla.
Calzadilla, excuse me. Let's see. Looks like you're sharing your screen.
Let me figure out.
That is okay. Right next to the leave button in the top right-hand side of your screen.
Okay.
There's a little square box with an arrow.
Okay. Can you see my
Yes.
screen? Awesome. Okay. Is it big now?
I see it. Looks like you're presenting and everything. So the title of Annalise's talk is The Better Side of Herbicide Development, a Rapid Method to Identify Transformed Wisconsin Fast Plants. So thank you very much. And the floor is yours.
Thank you. Hi, everyone. As Dr. Klocko mentioned, my name is Annalise Calzadilla, and I'm a student researcher in Dr. Amy Klocko's Plant Genetics Lab in the Department of Biology at the University of Colorado, Colorado Springs. Thank you for listening to my presentation on the better side of herbicide, developing a rapid method to identify transformed Wisconsin fast plants. So today I will give you a bit of background on the plants we are working with, talk about our goals and methodology, and discuss our use of herbicides and inform you of our current results. I will also speak about where we are in the plant transformation process of the experiment. We will finish up by talking about our future plans and the conclusions we have made thus far and give a thank you to those I would like to acknowledge. Wisconsin fast plants, or study plants, are known as Brassica rapa and are a cousin to broccoli. They are an ideal research subject because they are fast growing and can give quick results for both research and in the classroom. As the plants are related to a food product that people already consume, the information we gain from this model plant could one day be applied to crop plants. This is of essential value because every day 25,000 people die from hunger-related causes. A fruitful option to solving this enormous issue is to make genetically modified crops. The method we are developing will help this process of identifying these properly modified plants go smoother. Imagine this. You insert your favorite DNA into your favorite plant. You get seeds, you put the seeds in a petri dish, and now you have a bountiful array of plants. How can you tell which organism has your favorite DNA? They all look the same. Traits such as increased nutritional value or pesticide resistance are not going to display a markedly different visual appearance to the naked eye. By inserting your favorite DNA with a heritable trait, such as herbicide resistance in this case, when you plate the seeds on media with herbicide, you can see which plants have been modified because the modified plant will be healthy, both in size and coloring, while the others will be smaller and take on a sick appearance. The question is, which herbicide is best and how much is needed? This leads us to the root of our experiment. We are determining what the best way to identify transformed Brassica rapa is and see which herbicide is the best. Currently, intensive labor and in-depth analysis is needed to identify the plants, and typically only 1 in every 1,000 seeds have even been transformed. We are finding a solution to that issue. Along the way, we also need to determine which seed sterilization procedure is best. The methodology of our experiment can be broken down into these 4 steps: having a sterilization procedure, determining an effective herbicide and its concentration, transforming the fast plants, and finally, the pinnacle of this research, plating of the seeds to determine selection of the properly transformed plants. I previously completed a literature review due to the generosity of the LAS Student Summer Research Creative Work Stipend, and I have listed some of the key articles that have helped us develop the experimental procedure. The first article presents a method for transforming Arabidopsis thaliana, which is a cousin to Brassica rapa. This particular paper has helped with this experimental development. The second article was the paper that initially piqued our interest in the area and started this scientific project. The final article I listed here gave a method of identifying transformed plants that are a cousin of our study subject. Again, Arabidopsis thaliana. On this slide, I wanted to demonstrate how essential it is to sterilize seeds before plating them. Seeds often come along with fungal and bacterial hitchhikers that when grown in soil are of little consequence. However, these opportunistic friends are quite thrilled to grow when this plant's on the agar, which ends up inhibiting plant growth as seen in the bottom picture. When they have been sterilized, we can see how much happier the plants are. For Brassica rapa, we found that bleach sterilization was superior to an ethanol sterilization. The first herbicide I tested was kanamycin. These pictures are the results of the kanamycin levels on non-transformed fast plants. These plants had their pictures taken 3 days after they were plated on the agar. Visually, we can see a large difference between the plants that are exposed to 0 kanamycin with their leafy green foliage and developed root system, in comparison to the plants that have been exposed to 600 micrograms per milliliter of media. The high amounts of kanamycin cause the plants to be short and less developed, and have a purple and yellow color, which are a sign of stress. These results are what we were hoping for, a level of kanamycin that leads to a clear impairment of plant growth. Bostel was another herbicide whose effects were tested on Brassica rapa. The pictures of the plants were also taken after 3 days of being planted on Basta-infused media. At level 0 of the herbicide, the plants are healthy and happy, whereas at 400 micrograms of herbicide per ml of media, they are aggressively unhappy. In comparison to the level 0, the plants are much smaller. We do not see a huge color change in them if we only look at the top of the plants, which is what these pictures are showing, but on the underside of the plants, it is highly discolored as they're a deep purple. When we tested hygromycin as an herbicide, we ran into a different situation. These pictures are from exposing the plants to hygromycin-infused media, and are also after 3 days of plating. The fast plants still have no issue growing without the herbicide, as they display their intricate root systems. Although there is a difference in the plants after putting them on media with 50 micrograms of herbicide per milliliter, we don't see as strong of an effect as we were hoping. The herbicide caused the plants to be smaller, but they were still mostly green and had a decent size. At this point, we knew that unlike the other herbicides, further testing of higher concentration needs to be completed to see a more defined difference. Here are some of the images from the second round of hygromycin testing that we did with the larger concentration levels. After 3 days, we can see that this higher level is far more effective. The plants being grown on 100 and 200 micrograms of herbicide per milliliter of media are quite small, and the color is very poor, being yellow with tinges of purple. Increasing the herbicide to these levels had the effect we wanted on these plants. These two graphs are showing a graphical representation of the different effects on the plant's health depending on the concentration and the type of herbicide after 3 days. Plant health was scored on a scale of 0 to 4. The legend explains what these numbers mean, where 4 is referring to happy and healthy plants, and 0 is very impaired growth. These values given are based on an average of 2 plates for each level, and the conclusions are based on visual examination. This was important because part of our criteria for knowing if the herbicide level is sufficient enough is if the plant looks right. For example, when looking at kanamycin herbicide, it caused an underdeveloped plant organism, including the root system, but also had a large effect on plant color. The hygromycin mostly only had an effect on plant size. From these graphs, we can see a pretty clear inverse relation between plant health and herbicide levels for both herbicide types. When looking at the charts between the 2 rounds of hygromycin, we can see a big difference. Based on the first chart, there is an inverse relation, but is not as strong of a correlation as we would like. These results are suggesting we need to test higher amounts. After looking at the hygromycin chart 2, which is the one below, we have a more extreme drop than in the first at the beginning, but that is because in the first chart, we have levels between 0 through 50 in increments of 10, whereas in the second round, we have level 0, where the plants should be perfectly fine, but then jump in increments of 50, which is causing that large decrease. However, we can still see in that second graph that the more hygromycin is causing that larger effect we were hoping for. Now that we have completed our herbicide testing, we have to ask what is next. We have to begin the process of transforming the plants to have the genes we want. To do so, as this infographic is telling us, we need to encourage our plant cells to take up DNA, the DNA we specifically desire in this case, from the environment, and then make sure it is physically incorporated into the genome. Since it is not easy for plants to just do this naturally, we require the assistance of a specific bacteria called Agrobacterium tumefaciens. Now that we start out with a plasmid containing the genes we want to see, in this case, herbicide and any other favorite gene you may have, along with the necessary components that allow us to incorporate the plasmid into the DNA of Agrobacterium tumefaciens. The reason why we use this bacteria is because it is a natural genetic engineer for plants. When this bacterium affects plants naturally, it causes them to develop a disease called crown gall, which causes abnormal growth on the plant. Scientists have now been able to utilize the machinery in this bacterium to, instead of inserting crown gall genes into the plant, tumefaciens can insert many different kinds of genes, and therefore it can cause many phenotypic differences in these plants. Once the plasmid is in our bacteria, we introduce the bacteria to plant cells, causing the tDNA to be integrated into the plant cell, transforming it. This leads to us obtaining our desired transformed plants. To actually introduce the bacteria to the plant, we grow plants to the point of just before the flowers bloom, and we take the transformed agrobacterium and perform a process called the floral dip procedure. The floral dip procedure is exactly how the name implies, of dipping the flower buds into the bacteria. From there, the flowers need to be pollinated and then have to create seeds. These seeds from these dipped plants are what are going to be placed on the herbicide media to determine if they have been transformed. To actually introduce the bacteria to the plant, we grow, oh, my mistake. As mentioned, this procedure is not just allowing us to create herbicide resistance, but insert genes of our choosing. In this case, our favorite gene that we will be inserting is the GFP gene. This gene will also give us an additional visual marker of transformation. When held under UV light, transformed plants with the GFP gene will glow green. We also chose this gene because we are hoping to promote these plants and procedure for their benefit in the classroom. Realistically, what student wouldn't want to make plants glow? It's a very cool visual indicator for students to see. Of course, this procedure has the potential to help induce and identify other valuable traits suited to the researcher's desires. For the future, we will finish completing the process of bacterial transformation and the obtainment of transformed plants. We will collect and analyze the results of transformed plants on herbicide and the best method for selecting the transformed plants. After that, we hope to begin the process of publication. Our main conclusions today is that we have found an effective sterilization procedure. We have determined that kanamycin, hygromycin, and basta are all effective herbicides for Wisconsin fast plants, and have taken it a step further and determined the ideal concentration for these herbicides. I would like to give my thanks to those who have made this research possible, including the new Lab Startup Fund from the College of Letters, Arts, and Sciences, awarded to Dr. A.L. Klockow, the 2020 LAS Student Semi-Stipend, the 2020-21 LAS Student Faculty Research Creative Work Grant, and everyone within Dr. A.L. Klockow's Plant Genetics Lab. Thank you all for listening, and if you have any questions, please let me know. And that's the end of my presentation.
Clap, 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 clap. Anyone have any questions from the audience? Annalise, that was very nice. Do you feel that maybe you could find another like intermediate range around for hygromycin, like around the 300 to 350 or so? Because you killed them all off at 400, and you had a big, large jump from 200 to 400. Is there a way to potentially maybe
Yeah. Yeah, you're totally correct. That like 400, although it did probably annihilate our plants, would truthfully probably be too strong, because even when inserting an herbicide resistance, it's still only resistance. Like if you, for example, if you threw a herbicide resistant plant inside a bottle of kanamycin, it doesn't matter, it's going to die.
Mm-hmm.
So at the 400 level, we agree that it is definitely too strong. So we are more interested in that like 75 to like maybe 125 range to really get probably that more ideal herbicide level of being able to kill off or at least severely weaken any plants that are not transformed, while not actually killing plants that have transformation and you just gave them too big of a task to overcome.
Mm-hmm. Have you considered putting in I guess my original question was like the 300 range, but I guess, do you think that's going to be too strong?
I would assume that if I'm going to scooch back. So at the 200 level, which we can see right here, they're definitely looking pretty hurt. They're not enjoying much of anything. So it could still work at 200, but I do think probably at 300, it would be excessive. Okay. It probably would not be beneficial to them at all.
And then for your transformations, I mean, I agree that's gonna be a great experiment to do, and certainly a way to encourage student interest in this, because you get to see some cool green plants.
Yeah.
Are you just considering putting in just GFP under a constitutive promoter, but not having any unique localizations or anything like that? Or have you considered fusing GFP to something else?
By fusing it to something else, what do you have in mind per se?
Like another protein, making a C-terminal fusion. So you have your protein of interest plus GFP next to it, which if that known protein goes to somewhere else, it might be a cool thing to see, like instead of just the plant's entirely green, but maybe you then start seeing the vascular system or just the leaves or something like that.
Yeah, I think, well, I will say that hasn't something we have tested yet, but I think that is a great potential to see, and it's a very interesting way to tell the path of plant growth. I think with this procedure, when thinking of the students, it's more of since these plants are fast growing, you can get results in a much shorter time than with other plants. And so we wanted to it was more of an introductory to introduce the idea to students without perhaps making that like having to make that leap, right? And of course, by trying to attach it to a protein, that just adds in more chances for error, which I feel might be disappointing for a student when their plant doesn't do anything at all. Right. But I think that has actually value in studying as well. So I'm really glad you brought that up, because I think that would be a very interesting thing to see. And if we could have a procedure for that, I mean, that'd be excellent.
I think it would be cool to, fuse it to some, unknown protein and see if you can find some unique localization, or something like that. I think that'd be fun to, especially getting the students interested. Obviously, there would be that extra cloning step, because you have to clone your gene of interest to that GFP and then introduce it. And yes, as you rightly note, that could introduce error into it, especially if it's in a lab setting with undergrads, meaning you only have 16 weeks to do it. That could be tough, but maybe targeted genes could have already been cloned, and then you just introduce them, which I think would be cool too.
Yeah.
Cool. Does anyone else have questions? Any other questions from the audience? Okay, let's thank Annalise again.
Thank you, guys.
And so we'll move on to our next presenter. Our next presenter is also from Amy Klocko's lab. So our next presenter is Haley Klemp from UCCS. And the title of her talk is Analysis of Perfluorinated Compound Presence and Impact on Tree Growth in the Fountain Creek Watershed. And so, Haley, can you share your screen, please?
Yep. There we go.
And I believe you had a pre-recorded presentation, at least that's what I've been told, so.
I was going to do that, but my computer was acting weird when I was trying to do that, so I'm going to do it live instead.
Oh, okay.
Here we go. Okay, well, hi everyone. My name is Haley Klemp, and I am an undergraduate student attending UCCS. I am also in the Dr. Amy Klocko's lab, and today I'll be talking to you about the analysis of perfluorinated compound presence in the Fountain Creek watershed. So there are a few topics I will be going over with you today. First, I will delve into some background information regarding PFCs and why their presence in our local ecosystem is such a concern to us. I will also be expanding the areas of research we are most interested in focusing on, the experimental methods we utilized to determine the effects of PFCs in our local ecosystems, the description of the sites we chose, and the tree species of interest that we decided to focus on. I'll also be going over some data that we have collected thus far over the last 2 years, and also talk about the experimental errors and hurdles we have had to overcome. And to close, we will discuss some future goals that we believe will contribute to our research project. Now for some background info. So PFCs do not naturally occur in large amounts, but they are used often in the textile industry to produce fabric that is waterproof and stain or grease resistant. While the USA does not chronically use PFCs, there are traces in the fire suppressant foam sometimes used in training. So if you haven't heard already, back in 2016, there was a large spill when the Peterson Air Force Base released around 150,000 gallons of water containing this fire suppressant foam into the Fountain Creek watershed. The impacts the spill may have on our local ecosystem is still unknown, unfortunately. So we are interested in tackling the question, do trees in the Fountain Creek watershed absorb the PFCs that are present in the water? And if so, does the presence of PFCs affect tree growth and overall health? In order to answer these questions, we will need to determine if PFCs cause variation in the characteristics of leaves, as well as quantify the levels of PFCs in the leaf tissue of 3 chosen species at both sites. We will also be analyzing annual tree growth to determine if secondary growth is affected by PFCs. Now for our experimental plan. In collaboration with Dr. Christine Bierman of the Geography and Environmental Studies Department at UCCS, we will be taking tree core samples to analyze the annual growth of one of our tree species. We will also collect leaf samples from our tree species of interest to quantify the levels of PFCs present in these leaves in collaboration with Dr. Chanel Owens of the Chemistry and Biochemistry Department, as well as analyze leaf characteristics such as area, density, and petiole length. So here I have an illustration showing the relative locations of our sites, as well as the locations of Fountain Creek and Monument Creek. Monument Creek, our low concentration site, flows into Fountain Creek, our high concentration site. I have also indicated with the orange star the location of the Air Force Base where the spill originally occurred. As you can see, Fountain Creek runs through a large portion of southern Colorado into the reservoir. So now I'll be going over our 3 chosen tree species. The smallest of our species, the sandbar willow, has an average lifespan of around 10 years, and due to its size may be a good indicator of PFC concentration. These trees resprout well from roots, and their water consumption is unfortunately unknown. Sandbar willows also have a mutualistic relationship with beavers, since the moist environment they thrive in is created by the dams that beavers build with their bows. And here we have our second species, the Russian olive. These trees have an average lifespan of 95 years and consume around 75 gallons of water per day. They are also very invasive to our native species, as they produce asexually by spreading stalks underground, and their seeds spread easily and are viable for up to 3 years. And now for our last tree species, the Plains cottonwood. These trees have an average lifespan of 70 to 100 years, but have the ability to live up to 400 years if they are growing in a healthy environment. These trees absorb an average of 200 gallons of water per day when mature and are a pioneer species. Pioneer species are typically one of the first species introduced to a new niche, which encourages more plants and animals to arrive. These trees have an important contribution to our project, as we will be taking our tree core samples from the cottonwoods to measure the annual growth. Now I will go over our data collection. We started by collecting 20 leaves from 3 trees of each species to quantify the dimensions. Plants under stress tend to produce thicker leaves, so density may be a good indicator. We then crushed the leaves into a fine powder for chemical analysis of PFC concentration. Tree core samples were then taken from our chosen plains cottonwood trees to analyze growth behaviors. Tree rings not only show age, but also history of natural disasters and the tree's health over time. Wide rings indicate good health, while thin rings may be caused by drought, overcrowding, or too much shade, among other things. So on this slide right here, we have 2 graphs showing the average leaf area of each species at both sites in both 2019 and 2020. So why is area a useful tool in determining the amount of stress a plant is experiencing? Well, plants tend to produce smaller leaves if they are growing in a stressful environment. We have not yet seen too much change for this characteristic, and it is still too early to make any causal conclusions. On this next slide, we have the average individual density of leaves of the species at both sites in 2019 and 2020. Density is useful in determining if a plant is under stress because individual leaves tend to be thicker in plants growing in a stressful environment. Here we see something interesting because while the average density of our cottonwood and Russian olive trees increased, the willow trees decreased. However, it is still too early to draw any conclusions. These last graphs I will show you are the average petiole or stem lengths of our chosen species. Petiole length does normally vary depending on mechanical variables such as wind, but since stems play such an important role in transporting water and nutrients to other areas of the plant, this may be a good indicator of a plant stress level as well. So there are still some work in progress as well as future goals we plan to focus on. Quantifying the levels of PFCs in samples of all species at both sites is still underway, but it has been confirmed that there are PFCs in both the water and soil in these locations. We have also had a few errors along the way as well, the largest being that beavers love trees. Since a beaver chose one of our cottonwood trees to chew on, we had to choose a new tree. In the future, we hope to analyze tree growth pre and post spill from our collected tree cores and compare yearly precipitation during growing seasons to creek flow rates in order to rule out other variables that may contribute to stress. We would also like to expand the site to a larger area to cover more trees and to see how far the spill has traveled in our water systems. And that is it for my presentation. I want to take a moment to thank all of my fellow lab members who have helped with this project, and Dr. Amy Klocko for helping lead. I would also like to thank the Owens Lab for helping us analyze levels of PFCs, as well as the Bierman Lab for helping analyze tree core samples. And lastly, thank you to the LAS Innovative Research Institute for funding this project. And that's it.
Clap, 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 clap.
Thank you.
Very nice. I'll open it up to questions from the audience. Does anyone have any questions? Okay. Haley, I was struck by your data that showed it kind of looked like there were several times where especially in the Russian olives in terms of its service area, that even being grown in an area with more PFCs, it's actually they actually look like they did better. They had higher surface area. Would you say, yeah, that slide right there. If you can expand out your screen so we can look at the text a bit more, but or your PowerPoint. Yeah, so the Fountain Creek ones actually had a greater service area. Is it
Right.
Is it more likely that that has to do with their location? I mean, are they all sort of west facing and maybe get a lot of sunlight, or does it have to do with, one interpretation could be that they are more or that the PFCs may promote growth in some way?
That's possible. It's still too early to really decide because
I mean, I agree, it's a little early with that, but do surface areas on leaves change drastically over time? Do they have a wide variety, or is there a large variety in leaf size from year to year?
I believe so. We would have to do some statistical analysis with that most likely to see if the change is large enough for us to believe that the plant is under stress, that there's something going on. But we'd have to do this for probably at least an extra year or so.
Okay. And then some of the other stats that you had showed that the petiole length and the density is better in the Fountain Creek area. So Fountain Creek's for the cottonwoods there, the Russian olives. I guess for most of the cottonwoods there. The Monument Creek definitely seems better for that. So that would suggest maybe it's, at least with that, like you said in your presentation, maybe greater sort of exposure to wind or something like that would increase the lengths there. So that could be an environmental thing as well. But is it possible that PFCs could be compromising those lengths, especially if PFCs are absorbed through the petioles and go into the leaves?
Yeah, I was thinking it may be a possibility since the petioles play a role in transporting the products that are made to the rest of the plant, as well as water from roots to all the leaves, that if the petioles are being compromised in some way, we might be able to tell from their length to determine and see if the plant may be under stress. Okay,
interesting. I mean, that certainly could be an effect as well. And I'm just not sure. I just don't know. I'm very naive about some of those tree growth categories within that. So no, that's very interesting. Are there different water flow rates in those areas, or does Monument Creek and Fountain Creek have about the same amount of water flow in them? Because that could influence it too, not only in terms of the amount of the compounds, but just how fast they get water.
Right. If I remember right, I believe that Fountain Creek is usually higher.
Okay. Maybe because of those, like on your map there, you've got a couple other tributaries flowing into it, so maybe there's more in there. So
Right.
maybe water flow could also influence it as well.
Yeah,
Okay.
we hope to use the samples from our tree cores to compare past rainfall to see if maybe the plant was under stress because we didn't have really any rain that year. Like,
That makes perfect sense. There is a comment in the chat here saying that we did measure distance to water. That may be a factor as well. Okay, yeah, so the distance from the tree to the water source could also influence it. But I'm guessing that hasn't been fully, the stats haven't been calculated on that either. So that could be influencing it as well. So that how close the tree is to the water could also influence how much water it gets back out, which makes perfect sense to me as well.
definitely.
Are there any additional, Amy says not fully analyzed, any additional questions from the audience? Are there any additional questions? Okay, let's, oh wait, good presentation. Okay, perfect. So we have about a minute left to the next presentation. That'll give us some time to have it loaded up perfectly. So let's everyone give another round of applause to Haley. Very nice, great job. So Haley, if you can stop sharing your screen, that'd be great. Awesome. Thank you much, Haley. Our next presenter is James Duffy. James, if you can share your screen.
Yes, sir. Let me get that going. Let's see.
The little box with the arrow, you're getting it.
Yeah, let me see if I can get it playing at the same time. Should I just go right into it, sir?
It is 2 o'clock, so I'll introduce you first, and then we can, if you want to share your, put it on presenter mode and see if that works.
Yeah. Okay. Let me pause
I see it. Looks good. So our next presenter is James Duffy from USAFA. Thank you much for presenting and joining us today. The title of his talk, it looks like a poster, I should say, excuse me, is evaluating the performance of yeast across an elevational gradient, which is going to be interesting. Okay, James, the floor is yours. Thank you for presenting.
Cool. I will get going on that. Let me get this going. Can you hear the audio at all?
I hear you right now.
Okay, let me see if I can. There might be a little. Okay, let me pull out of this real quick.
Okay.
And
oh, it's a pre-recorded one. I'm sorry, I didn't realize that.
It is a pre-recorded one. Okay, I have to get rid of my headphones, and I'll do it through my computer audio. See if that.
Oh yeah, you might have to play it through your audio. That might.
Okay, we will try again. I will go back to sharing my screen.
I'm hopeful next year we'll be in person. So.
I know it's a little funky, but you know it's what we got.
It's what we got, and it's working. So.
Okay. So stop sharing options, and I will share the screen to my yeast poster. Holy moly. All right.
It's okay.
1 second. Okay, second time's a charm. To this one right here. Okay.
I can see you. Let's try it.
Hi, good afternoon. My name is James Duffy. I am a senior at the Air Force Academy. I'm a biology major. My mentor for this experiment was Dr. Kelly M. Kuhn, and we were evaluating the performance of yeast across an elevational gradient. So Saccharomyces cerevisiae has been around for a very long time. In ancient times, you could find evidence of Saccharomyces cerevisiae in beer, wine, and bread making. It was thought to have been cultivated from the skin of grapes, and it can be found in the natural environment around the world. Today, Saccharomyces cerevisiae is still responsible and is the major species used in beer, wine, and bread making. Our goal was to assess the yeast performance using cell count across various elevational gradients found in the environment. And the environments that we used were sea level at 0 feet, 28 degrees Celsius was the average temperature, mid elevations at 7,000 feet here at USAFA at 21 degrees Celsius, and high elevations at 14,000 feet, 18 degrees Celsius, similar to that of Pikes Peak. And how we were able to do this, we were simulating the elevation within an altitude tent used there at the athletic department in the human performance lab, thanks to Miss Diana Bollinger for allowing us to do that. And this tent, what it did was it manipulated the composition of air by displacing oxygen with nitrogen and vice versa to simulate a hypoxic environment for athletes to train at. And in this case, we grew our yeast. We grew the yeast in water baths in order to simulate these environmental temperatures. As seen in this picture, we used these glass test tubes that we filled with yeast and YPD broth, which is also used, it's called yeast peptone dextrose. And there were 3 water baths in each of these trials at this altitude tent. And each of those temperatures correlated to 18 degrees Celsius, 21 degrees Celsius, and 28 degrees Celsius. And I started the timers. I tested these in the morning, and over the course of a couple hours, I took samples over the course of 12 hours. When I sampled them, I added formaldehyde in order to cease the cell growth and to preserve the collection until I was ready to count them later that day. These were the little vials that I used. These contained the formaldehyde and the yeast samples that I took from each of these test tubes. After collection, I was ready to count them. And in this case, we used the Countess 2, which is an automated cell counter, in order to quantify the number of cell bodies in each collected sample. This machine was able to automatically count cells, the yeast cells, in a pretty accurate method. So we were then able to compare these cell counts across temperatures and atmospheric compositions. So on this chart here, we see that sea levels versus cell count. There's a times for 8 a.m. and 10 a.m. We used these 2 times because in order for us to present this, that was the best way to do it in order to share the findings. And our results concluded that Saccharomyces cerevisiae at high elevations, cell counts were significantly higher than the lower elevations of mid elevation and sea level. At mid elevations, we saw the smallest cell counts for the cold temperatures, as seen here. At sea levels, we saw the lowest not the lowest, but low levels across all cell counts of temperatures. And this surprised us because initially we predicted that this was where Saccharomyces cerevisiae was going to perform the most optimal. Being that it's the lowest stressed conditions, we were expecting high elevations to perform lower than the sea level, and it was the complete opposite, which was very surprising. So this data shows us that high elevation, specifically the medium temperatures as well, have the highest performance in regards to cell count, but we can't quite conclude what specifically happened. We don't know why specifically that this happened, but we can speculate that potentially there can be relation due to the altitude tent's increased nitrogen with the oxygen and nitrogen displacement. It may allow larger pools of nitrogen to be uptaken by yeast from the atmosphere. So further studies should be made to conclude why this happened. And if we were to do this study again, the use of a more advanced altitude chamber would be highly recommended. One that would able to artificially simulate not only the contents of the air, but also the pressures in a more complex and advanced way than what we had with the altitude tent. That way we could get a more accurate representation of the natural environment. So this study is important because we rely on yeast around the world. There really is no limit on how we can use yeast. Our little eukaryotic friends are important in culinary applications as well as industrial applications for making insulin, ethanol that is used in many things. Here in Colorado, in our very own Woodland Park, there is a brewing science institute where they study and cultivate yeast, and they have a bank of species of yeast for the use of brewing. But at high altitudes, such as Colorado, we can see that there is an influence, no matter how small that this study can show, on elevation and yeast and how they perform. Knowing more about yeast and how they perform at these elevations would offer more insight, not only on how to optimize yeast and their applications, but also to understand more of the life histories of all eukaryotic, of our eukaryotic ancestors. As a learning and developing scientist, this experiment has showed me a hands-on display of the scientific process. Dr. Kuhn and I were able to tackle the question about yeast by designing this experiment that would somehow isolate specific factors in the yeast's environment, such as temperature and air composition, at least. While there are more improvements that we can make to this in the future, we were able to do what we could with what we had. And after collecting this data, we were able to chew on this data to somehow propose possible explanations by reading scientific papers where we learned about yeasts and their ability to have nitrogen uptake from the atmosphere. So
that is basically it. Let me see if I can stop that going. Can you still hear me? Anyone?
Yes, we can still hear you, James. Very nice. Can you still share your screen? I'm really fascinated about your results. That was interesting.
Yeah, I thought it was far out.
Yeah.
Let me see if I can get that back in there. Sorry, I'm not a.
No, it's fine. It's fine.
Teams Pro. All right, is that sharing right there?
Yeah, it is. Because that was just really fascinating that the sea level ones are growing worse. And you say it in your presentation that that was unexpected. And I would have also said that too. Actually, I should have asked the group here. Are there any questions of anyone else? Does anyone have any questions before we go ahead and start? That was very nice. So.
Thanks. You're too kind. You're too kind.
Okay. I don't see any questions in the chat. So I was gonna just ask a few questions. Is it possible? So obviously at the sea level environments, you have greater levels of oxygen, and obviously you said that your chamber had your growth chamber was able to vary oxygen, but not necessarily pressure. Was that true?
Yeah, it's true. It was just so the machine would be able to control the composition of the air, but that solely. So
okay. Yeah, pressure is definitely, I could see that being an issue too. So I agree with your future directions of doing that again. But could it be that you're inhibiting cell growth maybe because there's too much oxygen? Maybe you're getting like reactive oxygen species or something like that.
Oh, totally. That could definitely be one of the proposed ideas where we'd have to further investigate that. But that's definitely on the table for options on why this happened.
Or yeah, reactive oxygen species, or even like you have greater oxidation of other compounds, aberrant oxidation. So growth at the higher levels are actually better in terms of that. I mean, that could certainly do it.
Yeah.
Did your cells? I'm sorry. I didn't mean to interrupt.
Oh, another possibility could be that maybe we weren't our measurements weren't close enough, and maybe there was more ethanol or different types of things that could have inhibited cell growth in those environments as well. So that would also be another attack again to see if that could be it.
That was actually just my next question, because obviously the oxygen levels in this can influence fermentation rates. If you're limiting oxygen, you're going to get fermentation. But also, oxygen is the final acceptor on the electron transport chain in these fungi. If there's varying levels of that for that final oxygen acceptor, maybe that would influence their growth rates. Like if you don't have enough, you're going to slow down the electron transport chain, slow down that flow of electrons to prevent essentially those compounds to be made, essentially preventing NADH to being oxidized back to NAD plus, or preventing the electrochemical gradient from being establishing, so you don't get as much ATP synthesized.
Yeah. It definitely can be one of the things. The metabolic activity in yeast, they can both do aerobic and anaerobic.
Right.
So when there is a limiting factor of either oxygen, whether it's present or not, it has the choice to do either depending on the environment. So that's another thing we'd have to really, in order to study that, we would have to come up with a very intricate, I don't know, that I'd have to really think about how to do that one.
Right. There are ways to monitor oxygen consumption. So in my biochem classes, I've actually introduced publications where researchers have monitored oxygen consumption. I just don't know the machinery or what's required for that. But if you monitor the consumption of oxygen over time in those chambers, so maybe you could see changes in your oxygen levels based with your cultures. Maybe that would give you a clue as in whether those cells are really consuming oxygen at a high rate or at a lower rate, which would be suggestive of different rates of the metabolic flux through the electron transport chain.
Yeah, like you'd be able to at least have some observational numbers there. That would be a way to do it.
It would be suggestive, at least.
I think that this performance of yeast is just super interesting, and And there's definitely an avenue to continue on with it for sure.
You can maybe even monitor NAD plus or NADH ratios. Maybe that's influenced. So the action of the citric acid cycle ahead of that would be fascinating too. It's just, that's a cool, I've always wondered that. Do you get subtle changes in the growth rates? And clearly we do.
Yeah.
On...
Yeah. No matter how, I can't say how conclusive this is. It's just a little intro into it, if anything. But there aren't many studies on the elevational effects on these, yeast. Like I did not find any papers, which is why we went ahead and did this.
Mm-hmm.
So it's definitely an area that should be looked at more. And
certainly something that we're all concerned about, because we here in the Springs are at that mid-range, mid-elevation range, while if you travel down to Florida, you're going to be at the sea level range. So
yeah.
So certainly, I mean, we all know about. Certainly, I get winded every now and then. Walking upstairs here at this higher elevation. That's partially because I'm probably out of shape, but possibly the concentration of oxygen, it might be 5% lower here. Actually, what are the concentrations of oxygen here in the Springs versus sea level?
It would be a matter of just less density. I can come up with a number. I have to get back to you on that one. I don't have it off the top of my head.
No, you don't have to. It's fine. That's right. It's gonna have the same concentration, just less of it. You're right. Good point.
Yeah.
Yeah. Because the atmosphere is a bit more spaced.
Which is why the need for a more complex altitude chamber would be the most accurate, I think, for this study.
Cool.
But
I agree. I think it's a good start, a very good start. Are there any more questions from the audience? Does anyone have anything they would like to ask? Nothing else in the chat, it looks like. Okay, I'm not seeing anything else. So let's everyone give another round of applause to James. Thank you much.
Thank you very much. Thank you very much.
He's here all week.
Appreciate it, sir.
All right, thank you. Okay, our next presenter is Kaylee Wong Doloff. Did I pronounce that?
Yes, you did.
Okay, awesome. So Kaylee, can you share your screen?
Yep. Give me a second.
You hit the little box up next to the leave, but don't hit the leave button. But the next. I've done that.
Oh,
I've done that. It's not good.
Okay. Oh, do you see it?
Yes.
Okay, excellent. I still keep going. Is it time?
Yeah, please go ahead. The floor is. Oh, I'll introduce you. So our next speaker from UCCS is Kaylee Wong Doloff. She's here from UCCS, which I think I just mentioned, and in the lab of Radu Kaskaval, and in the math department. Interesting. So the title of Kaylee's talk today is Data-Driven Control of Disease in Spatially Distributed and Age-Structured Populations. And thank you very much for, well, that's what I had on my sheet. Your title's a little slightly different, but that's fine.
I got the order mixed up, I think. Whoops.
That's quite all right. It's quite all right. No pressure. Thank you very much. And Kaylee, the floor is yours.
All right, thank you. Yeah, so as mentioned, I'm Kaylee, and today I'll be presenting my project that I did with Dr. Kaskaval in the math department, data-driven control of disease in age-structured and spatially distributed populations, or the other way around. So we'll just briefly go through why we're doing this, then we'll talk about how we developed this model that we're using, talk about what we're doing right now, and then talk about what we plan to do next, and just other directions that the work can be taken in general. So first, infectious disease modeling has been a staple in ecology and public health for a while. It's become even more topical now that we're in the middle of a global pandemic, and models are one of the best tools that we have to understand how the disease spread is progressing and how we can control it. So basic disease models typically have a few limitations. First, we usually assume that movement is random, which doesn't necessarily apply, especially in human populations. And there are diseases out there, like COVID, that don't necessarily infect and kill the same proportion of individuals across different demographics. So in this model that we did, our goal was to incorporate age dependence and non-random movement into the basic disease models. So the very basic disease model that we use is the SEIR model. Basically, the way that disease modeling in general works is we sort everyone into bins, so to speak, and each bin tells us what relationship they have with the disease. So the susceptible bin is everyone who hasn't been infected yet. Exposed individuals have been obviously exposed to the disease, but they're not infectious yet until they move to the infected compartment, where they can now start spreading the disease to other people until they recovered and they're moved to the recovered bin. So this basic model is given by this series of differential equations that describe just at what rate people are entering and leaving each compartment. So here we have beta, which is the rate at which individuals are exposed. N is the total number of individuals in the population. Sigma, which you can see here, is the rate at which exposed individuals will become infectious. Gamma here is how fast individuals recover, and mu is the death rate. In this case, we're just assuming that the significant deaths are coming from as a result of the disease and not natural. Now, in human context, we sometimes researchers will throw in a few extra components just to account for the different ways that human populations will respond to disease. In particular, that is the addition of the hospitalization component. And in addition, since we're mostly using this for COVID, we also made two boxes for infected individuals. We have asymptomatic infected individuals and symptomatic infected individuals. And so this is the COVID-19 model that's been used by most government entities. Now, this one is basically the same as the basic SEIR model, a few additional modifications to account for the extra compartments. So now we have a few extra gamma terms where we, since we have to have people move through each infectious component before they can recover, and we have to have people potentially be hospitalized before they can recover. So beta is still our exposure rate. N is still the total number of individuals in the population. Sigma is still how fast exposed individuals become infectious. And so this is where it changes, because now when we go from exposed, we move into the asymptomatic infectious compartment. And the rate at which people leave that and move to the symptomatic compartment is determined by gamma 1. Now, once we're in the symptomatic infected compartment, you have a few options. You can either recover by yourself, which is determined by gamma 2, or you have to be hospitalized, which is determined by eta here. And then when you're hospitalized, you either recover or you die. So the recovery rate is determined by gamma 3, and the death rate is determined by mu. In this case, we're assuming that only hospitalized individuals with this disease are going to be the deaths that we're accounting for. So now for what we're adding, first thing is age. Why age? You probably know that in the context of COVID, older people tend to be vastly overrepresented in the death counts compared to the rest of the population. Numerically, the disparity is pretty big. In El Paso County, the 18 to 64-year-olds, which we refer to as the general population as far as COVID goes, make up about 62% of the population, and proportionally they make up about 69% of cases, while individuals who are 65 and older make up 13% of the population, and again, proportionally make up about 15% of cases. However, when we look at deaths, we'll see that people in the general population only make up about 10% of the deaths, while people 60 to 65 and older make up about 90% of the deaths, which is a very large disparity that we can't, that we didn't want to account for just with one blanket mu parameter. So our solution to this was to basically duplicate each of the bins that we outlined in the last few slides and labeled each of them with age groups. In our case, we had group 1 be 0 to 19 years old, group 2 be 20 to 39, group 3 be 40 to 64, and group 4 be 65 and older. And so now we just have susceptible groups for everyone, exposed groups for every age group, and so on and so forth. The next thing that we wanted to do was take care of the movement component. So we decided to consult the literature and use what was labeled as the impedance model by Sala, which is, and the paper that we got this from is in the bibliography. So in this case, basically the goal here is to take the population in each location, as well as the distance between different locations, and basically combine those to get a general estimate of how likely people are to move from one location to the other. So here, our parameters are, so P sub I is the population at location I, D sub ij is the distance from location I to location j, alpha is the overall probability of movement, and so then we have F sub ij, which is the total number of trips from location I to location j, which we get by taking the population at location I, adding it to the population at location j, and dividing it by the distance, and multiplying it by that movement probability. Then we have T sub I, which is the total number of trips from location I, and we basically just do what we did for F sub ij, but we take that for every location except for I and add them all together. And so then our pi sub ij is the ratio between F sub ij and T sub I. That was a typo, it should just be T sub I. And that'll give us the probability that some people will move from location I to location j. So this location component mainly comes into play as far as modeling the disease exposure goes. So we took the general I term that was in the earlier models and replaced it with this jumble of math here. So the first term here, the 1 minus alpha times I sub 1i plus I sub 2i, is accounting for the spread within location I, while this next term, which is the sum of all the pi sub ij's multiplied by I sub 1j and I sub 2j, gives us the movement of infected people to location I from other locations. And in this case, note that I sub 1i and I sub 2i, that's the sum of all infected individuals in a location over all age groups, since we're obviously not limited to between age groups as far as people spreading the disease goes. So now this is our final model that we ended up using. So the updates here are obviously the location component that we added. And so we also updated beta, so that beta also depends on location. And death rate now depends on age. Also, we updated N here so that it's the population in each age group at a given location, since, again, each of these bins represents a different age group. So what we're doing right now is we are working on getting this model to work as applied to the COVID-19 situation in Colorado. So for our location data, we are wanting to consider two different scales, so to speak. So we have the large scale, which we're working with right now, and then ideally we'd like to scale it down to our smaller scale. So the large scale is made up of 11 regions, which are just made up of clusters of counties that are generally related geographically. And then on a smaller scale, we'd like to only consider counties to make it a bit more precise. So we're still working on actually getting our parameters to fit with the data, but we decided to take all the populations from our regions and throw them into the model to look at how just movement generally tends to affect how the disease is spreading among Colorado. So on the left here, we have less movement between regions. So we set alpha to 0.1, which means that people are very 10% likely to move between different regions. And then on the right, we have alpha equals 0.5, so people are 50% likely to move around to different regions. And so just to get a better look at this by comparing two regions with different populations, whoops, that was the wrong way. Here we have the graphs for the Denver metro area and San Luis Valley on the right. And so obviously the Denver metro area is massive, while the San Luis Valley comparatively does not have as many people in it. And so you can see here where when alpha is low, i.e. movement between regions is low, we see that Denver still has this very large spike in cases, while the spread in San Luis Valley is a bit more subdued. Whereas once we pump alpha up to 0.5, we see that Denver also, still has a spike, but it's not as noticeably changed. Whereas when we look at San Luis Valley, this spike here ends up getting much bigger compared to where it was when there was very little movement between regions. And this holds in general when we look back at the general graph for regions of smaller populations. So regions of larger populations tend to be generally look about the same, while smaller populations end up having a much bigger spike once we increase the amount of movement. So obviously, even though we were trying to compensate for some limitations in traditional disease models, with this one, we still have a few limitations that we have yet to account for. So first, movement can only really be modeled approximately. Just in general, human movement is still developing in the field of mathematics. So I expect that once that gets better, we could even continue to make this more precise. And we're still assuming that movement within location I is random. We also don't account for factors that could decrease spread, like vaccination or like lockdowns in the case of COVID, or increase spread, like that holiday travel spike that we saw in November and December, and weather. We also assumed that you go from exposed to asymptomatic to symptomatic to recovered, which isn't necessarily accurate. For example, with COVID, we know that people can not show any symptoms, spread the disease and recover without ever moving into the symptomatic compartment. And we still assume that immunity is permanent, which we're still not too sure about with regards to COVID. Finally, in theory, we could scale this model down infinitely, but realistic results rely on good data, and it gets progressively harder to find on a small scale. For instance, even at the county level, some of the counties are so small that you can't even find data publicly available for individual counties. So future work, our first priority is to turn our parameters into functions so we can actually get that seasonal spreading variation that's actually been present in the data. In particular, we are looking at making beta, the exposure rate, into a function by fitting it to case data. And then our ultimate goal with the model is to add in control mechanisms like vaccination, for instance, because obviously we would like to use the model to as a hypothetical thing to determine how much of a given measure you'd need to control the spread at a desired amount. More generally, we could also scale the location down further. Again, that would be more challenging given just the data availability, but it could be done. And theoretically even go down to model community spread. And then on the other hand, we would also like to take this and kind of blow up the scale a little bit and look at movement between states as well. And then finally, the age compartments that we used, even though we labeled them as age just for the sake of this project, they're pretty versatile, and you could use them for really any other demographic that you'd like to look at. For instance, like ethnic groups and gender. So yeah, that's pretty much it. I'd like to thank the College of Letters, Arts, and Sciences for providing financial support for this project, and I would also like to thank Dr. Cascaval for all his help on this project. So, and here's the literature we use, and that's all I have. So yeah, I guess that was when I open up the questions.
Clap, 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 clap. Very nice. Very interesting. I'll open up to the the floor first. Does anyone have any questions? And please, you're welcome to put them into the chat. I know I've got a couple questions. I don't see anything in the chat. So Kia, that was really fascinating. I'm really And I think I sort of answered my question a bit based on your future directions, but your movement modeling, human movement is really clearly very difficult to model.
Yeah.
So within your modeling for that, you're really just going from you're not really accounting like say if I go to the grocery store or anything like that. It's more that's what you call an intercommunity modeling, correct?
Yep. Yeah.
And then travel to other parts of the state. Is that what you're sort of modeling in the other part?
Yeah. So basically we're just accounting for that, like intercity travel and stuff, or I guess inter-county technically, since we didn't go that far down. But yeah, so that's one of the hard things about it, is that a lot of the even within the context of COVID, a lot of the spread that's been taking place has been within has been community spread, which has been, and yeah, it's pretty difficult to model. Like there's an entire subfield of math just dedicated to describing how
people could you model it based on maybe day? So I'm trying to think large gatherings such as religious services on Sundays. Could you go model it to that? So you can maybe see if there's greater spread in those large gatherings versus individuals who don't go, or there's less religious affiliation or something like that. Again, I'm not trying to be specific about that, but it just seems like that could be another large gathering that's related to a certain day.
Yeah. I think that would be a pretty cool direction to take it. Just as far as I guess parameters go, I think just the data aspect of that would be a bit more difficult, since obviously the model that we use is based on population, and obviously you can't really do that when it just comes to the grocery store and church and school and stuff like that. But yeah, no, it's totally a cool potential future thing, and I think it'll definitely become more viable, I guess, as just the field of movement modeling moves along.
Right. Because that's I can't even imagine trying to model that with variables like that. That just seems so okay. It blows my mind, but I can't do it. And then how close are your models to? I know you say it doesn't really fit, but is it looking pretty close? Like you could get it estimating the transmission rates or the data that you're getting from El Paso County?
Yeah, so that's actually what we're working on right now, is trying to get that to fit, because right now our parameters are kind of rough estimates right now, and so we're hoping to be able to fit that to the graph more closely.
Okay, it was fascinating. I'm wondering if you could just go down to I don't know, thousandths or 10,000ths place to see if you get into the modeling effect or something like that, rather than just 0.1. So, but anyway, hey, that was a great talk. It is 2:40. If there are any other talks, please let me know. Sorry, of course there's other talks. Any other questions? Okay, let's thank Kaylee one last time. Thank you very much for a very nice talk.
Thank you. Thanks for listening.
That was very nice. You actually made math enjoyable for me again. So that's a joke, Radu. That's a joke. All right, let's move on to our next presenter. The next presenter is Chaz Fields. Chaz is here. Chaz, can you share your screen, please?
Yeah. Let me share content. Oh, hold on. I'm sorry. Apparently, I need to allow this in my my computer. My bad. I'm sorry.
Well, that's okay. Take your time. There's no rush. Good old technology. Good old pandemic.
Yeah, I think I have to
next year we will. I'm sorry?
I think I need to close Teams real quick, it's saying. So I'm going to do that and open it again. I'm sorry.
Okay. Are there any, while we're waiting for Chaz to exit and return, are there any questions that anyone has about the meeting, about Kaylee's not on the spotlight anymore, but are there any questions in general about other CSURF aspects, any other life in general questions? What is my favorite food? Man, that's a good question. Open up to the class, Open up to the meeting here. I would have to, man, the real questions. That is, God, I like so many foods. I don't have a good answer, Annalise. I don't have a good answer. Just everything.
Okay. I think I'm back. I think I'm good
Okay.
now. Okay. Again, I apologize for that.
No, it's fine. It's fine. Pizza is really good, though. I agree.
Okay. Okay, can you see it?
Not yet.
Oh gosh. Why am I so bad at this?
It's okay.
Share.
The
Okay,
share.
can you see it now?
Sometimes if you share your entire screen, instead of just the presentation, it might work.
Okay, I thought that's what I was doing. It says I am sharing, but you can't see it?
I don't see anything yet. No.
Oh gosh.
Okay. I don't see it.
Still no, huh?
No, I don't see anything yet.
Oh my gosh. So I just to clarify, I hit the share content and then I hit screen.
Either screen or your actual, sometimes it might be actually on your presentation if you have a PowerPoint open.
Okay, let me try it that way. Can you see it now?
No.
It says
sometimes
that
it takes a second to load up. But
okay. Of course I would have these issues. I apologize. I don't know why.
Don't feel bad. It's okay. It's quite all right.
It says it's presenting right now, but you can't see anything.
I can't. Can anyone else? Annalise says no. Is it on a different? Wait, there it is.
There it is?
Yeah.
Okay, I guess I just had to do it like
Teams right now. I
6
see the chat in Teams.
Okay, can you see my PowerPoint now?
Yep.
Okay. All right, sounds good. All right, I apologize for all of that, everybody.
Don't worry about it. You got this.
So, hello, everyone. My name is Charles Fields, or Chaz Fields. And I'll be presenting my research project entitled Elucidating Factors That Can Impact Public Policy Interventions from the COVID-19 Pandemic. This project is funded by a rapid grant from NSF, and it was conducted by myself and Professor Lisa Hines, with collaboration with Philip Brown, who's the principal investigator, and Gia Barbosa. So the SARS-CoV-2, the coronavirus responsible for the COVID-19 global pandemic, was first identified in Wuhan, China in late 2019. Shortly following its emergence in China, the first confirmed case in the United States occurred on January 20th, 2020. As of December 31st, 2020, an estimated 19.66 million cases and more than 330,000 deaths were reported in the United States alone, making it the most severe epicenter for the virus. Incidence and mortality rates vary greatly across states, with Texas, California, and Florida collectively representing nearly 30% of all the reported cases in the country. Irrespective of this variability, COVID-19 disproportionately affects the elderly, minorities, and those of lower socioeconomic status. We focused our study to 10 states that would best represent a wide socioeconomic, demographic, and geographic range, while also covering the spectrum of COVID-19 burden. These states included Alabama, California, Colorado, Florida, New York, North Dakota, South Dakota, Tennessee, Texas, and Washington State. This graph here depicted shows the number of new positive COVID-19 cases per 10,000 population per week for each of the selected states throughout the year of 2020. Reviewing the graph, we see 3 distinct waves of increased incidence throughout the year. Interestingly, the burden of incidence experienced by each state changes throughout each wave. For example, If I can draw your attention to the light blue line that represents New York, we see that above all other states, New York experienced a heavy burden of COVID-19 incidents in the earlier part of the pandemic, or what we can think of as the first wave. However, following the introduction of strict social distancing policies, New York's COVID-19 incidence sharply declined and remained rather low even throughout the second wave of the pandemic. So now changing our focus to the second and third waves, we see that Florida, depicted by the yellow line, experienced a drastic increase of burden throughout the second wave, while North and South Dakota, which are the green and dark blue lines respectively, experienced the heaviest burden throughout the third. So for this second graph, this depicts the number of COVID-19 deaths per 100,000 population for each of the 10 selected states throughout 2020. Akin to the first graph, this graph depicts 3 ways of increased mortality throughout the year. Again, we see the burden of mortality experienced by each state changes throughout each wave. Perhaps not surprisingly, the burden of COVID-19 mortality experienced by each state follows closely to that seen in the previous graph depicting weekly cases, meaning states like New York, who experienced a high burden of COVID-19 cases in the early part of the pandemic, also experienced a high burden of COVID-19 related deaths during the same period. However, both graphs that we've been studying depict COVID-19 incidence and mortality data at the level of the state. And although this data is useful, within state socioeconomic and demographic variations make enacting preventative policies difficult. Therefore, it is vital to address the problem at a more granular level. So extensive efforts have been made to elucidate factors that contribute to disparities among individuals. However, public policies are implemented at a state or county level, and little research has been done to investigate factors that impact disparities at the county level. So in order to achieve more granular and focused efforts to understanding the factors that most heavily impact the community burden of COVID-19, we use publicly available census data from the US Census Bureau, the CDC's COVID data tracker, and the CDC's Social Vulnerability Index data hub to identify possible county level predictors of COVID-19 incidence and mortality. Through these resources, we identified 11 variables of interest that would be most likely to act as significant predictors of incidence and mortality at the county level throughout the 10 states of interest. These predictors include the percentage of the population that's over the age of 65, the percentage of the population that is Black, the percentage of the population that is Hispanic, the percentage of the population that is male, percentage of the population that is uninsured, the mean unemployment rate throughout the year of 2020, the population density of the county, which is measured in the number of people per square mile, the median household income, the percentage of the population that is registered as Republican, the percentage of the population that is obese, and if the county experiences high level of tourism. So we began our analysis by constructing a fully saturated multivariate linear regression model for each of the selected states for both incidence and mortality. And then using each of the saturated models, we then performed a backwards elimination regression to gradually eliminate variables from the saturated models until a reduced model was created that best explains the data. Predictors remain in the model if they met the inclusion value of alpha 0.05. So I would like to note that we chose a backwards elimination approach in order to preserve any collinearities between predictive variables. And for each regression model, the AIC residual versus fitted plot and the QQ plots were then assessed to ensure proper fit, and the results are as follows. So table 1 displays the results of the stepwise regression models assessing the significant predictors of COVID-19 incidence across counties at the 10 selected states. So to better familiarize you with the data, let me focus your attention to the left side of the table. Here are listed the 11 predictor variables that were chosen for analysis. Each row represents a single variable. To the right, we'll see a column for each state, and within each column, you'll see either a blank space or a colored arrow. The blank spaces indicate that the variable is not a significant predictor of COVID-19 incidence at the county level for that specific state. A space that contains a green upwards facing arrow indicates that the variable is a significant predictor of COVID-19 incidence at the county level for that specific state, and that the correlation is positively associated. For example, within Colorado counties, the greater the percentage of the population that is Black, the higher the expected burden of COVID-19 incidence. Alternatively, a space that contains a red downward facing arrow indicates that the variable is a significant predictor of the COVID-19 incidence at the county level for that specific state, and that the correlation is negatively associated. For example, within New York counties, the greater the percentage of the population that is over the age of 65, the lower the expected burden of COVID-19 incidents. So now that we might hopefully understand how to read the table, looking at it, we see that there's a fair amount of variation between what is considered a significant predictor of COVID-19 incidents between state counties. However, there are a few predictors that stand out as being significant across many states. The most conserved significant, I'm sorry, I lost my place. The most conserved significant predictor across the selected state is median household income. Our results show that all but one state has median household income as a significant predictor of COVID-19 incidence at the county level. And within these states, all but two represent a negative correlation, meaning a median household income increases COVID-19 incidence. As it increases, COVID-19 incidence decreases. This finding introduces many intriguing questions. For example, could higher household income be associated with a greater ability to conform to public policies? For example, stay-at-home orders, which limit income. Similarly, two other predictors that I would like to draw everyone's attention to are the percentage of the population that is Black and the percentage of the population that is Hispanic. Across the states, these two variables are the second most conserved significant predictors of COVID-19 incidence, and in nearly all cases represent a positive correlation. This finding suggests that counties with higher percentages of minority groups suffer from a greater burden of COVID-19 incidence. I'd like to note that for both variables, there is a single instance where the findings of the regression model found a negative correlation, meaning that a lower burden of COVID-19 incidence should be expected. In both of these instances, however, the variation between counties is very limited, meaning that they don't have these populations in their state or within their counties, and therefore the results might be like slightly construed. So in table 2, this table displays the results of the stepwise regression models assessing the significant predictors of COVID-19 mortality across counties of the 10 selected states. So this table can be read in the same manner as table 1, with the presence of arrows indicating significance and the direction and color representing the type of association. Similar to findings of table 1, table 2 shows a fair amount of variation between what is considered a significant predictor of COVID-19 mortality between state counties. Interestingly, the most conserved predictor across the selected states for COVID-19 mortality at the county level was a median household income. Akin to what was found for incidence, median household income was nearly always negatively associated with COVID-19 mortality, meaning that as the median household income increases, the burden of COVID-19 mortality decreases. Similar questions arise from these findings and point to a factor that should be heavily considered by policymakers when enacting policies focused at disease interventions. Another variable that was conserved across the selected states as a significant predictor of COVID-19 mortality at the county level was the percentage of the population that is obese. 4 of the 6 states that had this variable as statistically significant showed a negative correlation. This finding is rather interesting, as research conducted on an individual level of the risk factors of COVID-19 mortality found obesity to be positively associated. Such findings support the idea that conventional understandings of the significant predictors of COVID-19 mortality, as witnessed on an individual level, are not completely consistent at the county level. So finally, I would like to draw everyone's attention to the percentage of the population that is Black variable. Similar to what was seen for incidence, this variable was statistically significant for half of the selected states, and nearly all instances represented a positive correlation. Again, in the one that is showing a negative correlation, it's in a state that doesn't have very many African Americans in it, so please take that with a slight grain of salt. But these findings suggest that not only are counties with higher percentages of African Americans suffered from a greater burden of COVID-19 incidence, but also mortality. To conclude, our preliminary findings suggest that the conventional understanding of significant predictors of COVID-19 incidence and mortality as witnessed at an individual level are not completely consistent at the county level. Political party affiliation and mean unemployment rate hold little predictive power, while age shows an inverse correlation, at least when it comes to incidence. Instead, policymakers should consider demographic and socioeconomic factors. In light of vaccine rollouts, understanding the significant predictors of COVID-19 incidence and mortality at the county level will be of vital importance for future mitigation efforts. So some of our future goals for this project include further exploring the relationship between other significant predictive variables of incidence and mortality across selected US states. For example, some recent literature has suggested a correlation between COVID-19 incidence and colder temperatures. We would also like to generate similar stepwise regression models of predictive variables of incidence and mortality across other state counties. And finally, we'd like to work to better understand the underlying factors that lead to the unexpected results, some of the unexpected results that we saw. So thank you to everyone for taking the time to listen to the presentation.
Thank you very much, Chaz. That was very nice. Anyone have any questions from the floor? I know I'm seeing a couple of great jobs, which is great. Chaz, is there a correlation between county population size and of those states and COVID incidents? Like does it happen more in more densely populated counties?
So, and one of the predictors that we included was population density, which we hope would kind of help to answer that question. And looking at the tables, population density was one of the least significant variables. And in the states that actually did show correlation within their counties, it was actually normally negatively associated, meaning being, there would be less cases in these like higher dense locations, which I feel like I've kind of thought through a little bit because my brother lives in Denver. So when I go to visit him, it's vastly different when you go, he lives right off of 16th Street. So when I go to his apartment, everyone's wearing a mask outside. When I go down here, that's not really the case. So I think there's just stricter policy being enforced in areas that have higher population density that probably is accounting for that.
Yeah, I've heard it's even worse in like the South and stuff like that. Do like general areas of the country? I mean, I guess you kind of did that with your state level regulation. But I guess I was trying to think of like the South versus the Midwest versus the Pacific Northwest, that kind of thing. Would that be a way could you group them?
So I did try, again, I do hope to get more states. At some point, I want to have all the states and then do some other modeling to see what predictors, if I have every county in the US together. But with the 10 states I did select, I tried to pick ones that are in all geographical locations to get an idea of, different, basically in nearly all of the variables, try to have, like a wide range of, basically, like in South Dakota and North Dakota, most of I think the highest percentage of African Americans that they had in one of their counties was 2%, where in my Alabama one, I had some that were over 60. So I did try to include that. So I could see if there was any difference between like states for that reason. But I feel like from the results, it seems like for the most part, it's fairly consistent. I mean, kind of. There is variation, but for some of the like big predictors like income and, like I said, the percent of the population that's Black and Hispanic, like they kind of stay significant no matter what geographical location it is in. But I don't know if that really answers your question.
I think it does. Again, it's sort of, I'm going back to like Kaylee's presentation. I'm wondering if like percentage religious affiliation would do something like that too. I don't think that was one of yours.
It's not.
I wonder if that would be possibly correlated because people go to church on a weekly basis and they might be more exposed in those large group settings.
That would be an interesting, that might be an interesting variable to put in. I'll make a note of that for future work.
Okay. Any final, so thank you, Chaz. That was an interesting presentation. Any final questions or concerns? Questions from the audience? There should be no concerns. Questions from the audience? I'm not seeing anything else in the chat. Okay. All right, let's give Chaz another round of applause. Thank you very much. And then our final speaker of session 2, room 2, is we have two UCCS presenters, Lindsay Smith and Amanda Yu, which I believe you guys are both here. Yeah, okay, I see both of you. Okay, yes. Lindsay, looks like you're gonna share your screen. And
Amanda will be sharing her screen.
Oh, okay. Okay, perfect.
Can you see that?
Yes, yes. And so the title of their talk is Annotation of MAT Genes in the Yeast Stamera guercum and Candida rabinoformitans. I hope I got those scientific names correctly, even though I'm a biochemist. I think I got them. Thank you very much, and the floor is yours.
All right, thank you so much. Yeah, so hi, I'm Amanda.
And I'm Lindsay, and we are actually students from Colorado College.
Yeah, I think there was a little mix-up.
I'm so sorry that it's on my thing as UCCS. Thank you very much, Colorado College presenters. Thank you very much.
And so for our research, we focused on locating the MAT genes within different yeast species, specifically Stamera querculum and Candida arabinofermentans. And we did this with 2 other lab mates who were not able to present today, and with Dr. Sarah Hansen as our mentor.
So the purpose of our experiments was to further understand the evolution of mating type switching in yeast species. And the mating type region contains repetitive elements that are difficult to assemble using short read sequencing data. Therefore, we sequenced the genome of 4 yeast species using min-ion sequencing to obtain long read sequences, to then better annotate the MAT regions in yeast. This data will be used to assemble whole genomes in the future. We also focused on the purity and concentration of the DNA to select the genomes for assembly, to then identify major evolutionary changes. The NCBI blast search and the program Artemis were also used to examine sequences for the presence of MAT A1 and A2 and/or MAT alpha 1 and alpha 2 genes, which would help determine whether the mating pattern of the species were homothallic or heterophallic. So looking at figure 1, we see the zygote formation in yeast species undergoing mating type switching, and the figure starts with an alpha type cell, which then switches to an A type cell so that it can mate with another alpha type cell. Essentially, there needs to be an alpha and an A type cell in order for a zygote to form if the species is heterothallic or a secondary homothallic. Looking at figure 2, we see the 3 primary reproductive patterns of the yeast species analyzed, which are primary homothalism, secondary homothalism, and heterothalism. And with primary homothalism, any cell can mate with any other cell. For example, an A cell and an A cell, an alpha cell and an alpha cell, and an alpha cell and an A cell. The column on the left portrays the primary homothalism mechanism, with the top image showing a contiguous primary homothallic mechanism where the alpha and A-type genes are located on the same contig. And the image below depicts the non-contiguous primary homothallic mechanism where the alpha and A genes are located on separate contigs. The column in the middle exhibits the heterothallic mechanism where only cells of opposite mating types can mate, meaning only alpha type and A-type cells can mate. And looking at the middle image, if there are no A-type cells present to mate with the alpha-type cells, then mating type switching does not occur, and the image below shows that nothing happens if there are no mating type genes present. The column on the right depicts secondary homothalism, where cells can switch their mating type. As shown in the figure, there are 3 types of secondary homothalism, including flip-flop mechanism with 1 repeat, flip-flop mechanism with 2 repeats, and a 3-locus system. The repeats are inverted repeats, which are located next to centromeres and telomeres. And looking at the first image of the FF1 mechanism, the A cells are silent because they're located next to a centromere telomere, and the alpha cells are active because they are not located next to one. However, the cells can undergo the flip-flop mechanism where the alpha cells switch places with the A cells and therefore become silenced, and the A cells become activated. The black X in these figures indicate where the switching occurs, and the 3-locus system is when there are 3 mating type cells present, but only one of them is being transcribed while the other two are silent. This is known as a cut and paste mechanism where the A-type cells become active and the alpha-type cells become silent and vice versa.
All right. So in terms of our actual procedure, we kind of had three main parts. So we had our sample preparation, genome sequencing, and data analysis and processing. And so sample preparation, for that, we first cultured many different strains of yeast and extracted their DNA using a DNA extraction kit. And after that, we performed a DNA quality control assessment using nanodrop, gel electrophoresis, and qubit to check the quality, quantity, and purity of the DNA. Next, for the genome sequencing, we used nanopore sequencing, and we first isolated all the DNA and resuspended it in a smaller volume of solvent to have a more concentrated solution. We then added a unique barcode to each sample with for each species, and then we loaded all of the DNA onto the nanopore sequencer and let it run for 72 hours. For data analysis and processing, we annotated the yeast species genomes for MAT genes using BLAST, and we then treated and filtered our data that we got from our nanopore sequencing using command line with nanofilt and porechop, and we at the end we analyzed the quality of our data after treating our sequences with these different programs.
So figure 3 depicts our DNA extraction quality control results, and we found that 4 species had sufficient purity, quantity, and integrity of DNA to then proceed with the nanopore sequencing. The Starmera querculum and Candida arabinofermentans species had the highest quality DNA, as indicated with the 15.9 nanograms per microliter and 13.7 nanograms per microliter nanodrop quantities. We also found that these samples had bright gel electrophoresis bands of relatively large length, which was nice. And figure 4 depicts our 2 species with the highest quality of DNA. So the Starmera querculum had significant hits for the MAT A1 and MAT A2, as well as MAT alpha genes from A rubicins, and Candida arabinofermentans had significant hits for the MAT A1 and MAT A2 genes from A rubicins. The figure also shows what contigs these mating type genes are located on. And then going to figure 5, it's a table of our nanopore sequencing data, and we use the programs porechop and nanofilt to see what combination of the programs would produce the highest quality and length of reads. So we found that the chopped and filtered reads, which use both porechop and nanofilt commands, produce the highest quality and lengths of reads, as depicted with the 6,627 base pairs for the N50 read length, 4,517.3 base pairs for the mean read length, 3,116 base pairs for the median read length, and 9.4 for the mean read quality. So the last row on that table.
All right, and just to kind of recap, so the objective of this research was to search the genomes of Starmera querculum and Candida arabinofermentans for MAT A and MAT alpha genes. And so in Starmera querculum, we found that the MAT A gene, the MAT A1 gene, excuse me, was on a different chromosome than the MAT A2 and MAT alpha 2 genes. And this suggests that Starmera querculum is homothallic and can reproduce using A and alpha mating type cells. In Candida arabinofermentans, we found that the MAT A1 and MAT A2 genes were on the same chromosome and lacked a mating type locus on another chromosome, which indicated that this species is heterothallic. Some of the main takeaways from this are that these mating types of yeast indicate whether the species undergoes copy and paste or flip-flop mating type switching, and we can also use this information to determine the evolutionary development of mating type switching systems in different clades, and we could also use this to infer more possible evolutionary advantages to each of these mating type systems. Some future directions for this research would be to sequence the genomes of more yeast species and to determine the 3D arrangement of the yeast chromosomes. And this would allow us to have a better understanding of the processes and mechanisms of mating type switching, which we will be focusing on over the summer. And we also wanted to give special thanks to Dr. Sarah Hansen, who was our mentor throughout this, and the molecular biology department in Colorado College for supporting our research.
Okay, very nice. Any questions from the audience? Let's see, anything in the chat? No, I have not. Can't see anything in the chat. Okay. Okay, excellent presentation. So you guys just started the deep sequencing of that, the long read sequencing. How's the genome assembly working on that? Do you guys have the contigs put together in a good organization yet, or is that still ongoing?
I think that's still ongoing. We haven't really gotten to delve too deep into the actual data because we got that towards the end of our research. So.
Oh, sure.
Yeah.
That's understandable. I know you guys have if you have the block system, you don't have. Well, you got 3.5 weeks to get the data, so it's a little time crunch, but that's okay. Because that's exciting. Because Dr. Hansen and I are working on a collaboration as well, and we know that Hagalorum has a translocation compared to O polymorpha. So that's was there confirmation of that genome rearrangement in your species? I know you're focusing on the MAT regions, but does that?
No, we have not found anything about that.
Okay. Comparatively, okay. Let's. I'm sure that it could be in the actual data set a little bit, but that's fine. So how does so you said you found the MAT locus on a different chromosome for the one species. I can't remember what the species was. I guess the poster's been closed, but it began with star.
Starmer querculum.
Yeah, so that's on the MAT locus is on a different chromosome, or sorry, the MAT locus is on a different chromosome from one of the mating types.
Yeah, I think that was where we had let's see, the MAT A1 gene was on a different chromosome than the MAT A2 and MAT alpha 2.
Okay, so how do you know? Would you predict then that those genes would be able to interact, or that there could be some sort of regulatory pathway to allow mating type switching?
We really don't know at this point. That would be something interesting to look into. I don't know if Lindsay has anything to add.
I think we could actually predict whether it's homophallic or heterophallic. I don't have that figure pulled up if you have it, Amanda, but with the alpha and A, if they're on the same chromosome, I think they could still undergo that. And then also we could maybe hypothesize that they might go under flip-flop mating type switching, where one of the alpha or A's would switch to a different chromosome.
Okay. Yeah, I see it right there on the top left portion of your figure there. So you're saying it's a different chromosome for those two, so it suggests that they have different expression patterns.
Yeah, so for Candida or Abenofermitans, you only see A and you don't see alpha. So that suggests that it's either heterophallic and it would need another alpha cell, from another species to mate with it, or it could be homophallic, where it could switch essentially its A cell into an alpha cell, and then it could kind of mate with itself.
Okay. So it doesn't have the alpha gene at all, the Candida genus, at least the Candida species, I guess, just to say.
Yes, at least on that chromosome.
Okay, on that chromosome. Okay, but Sturmera does. It has the potential. So you're saying that could be heterothallic.
Yes, I believe so.
Okay. Does the fact that those genes, I've always imagined, maybe I don't know mating type switching as much as I should, and this is my own deficiency in the literature. I thought they had to be on the same chromosome, like in like mating type switching in C. elegans, not C. elegans, darn it, Andy, wrong species. Saccharomyces cerevisiae, excuse me. Since that switches, I thought the mat locus and then the two copies are right next to one another, and you can enter it in or enter either the A or the alpha in. I thought they had to be on the same chromosome. Does being on a different chromosome matter?
I'm not too sure. We didn't really see that in any of the literature we read. From what I understand, in this diagram, they should be on the same chromosome, but I am not entirely sure.
And the species I really look at mostly, or I have looked mostly at in the literature, has been Saccharomyces pombe and Saccharomyces cerevisiae. And I thought Pombe's, it has some heterochromatin formations on one chromosome, and both copies are on one chromosome. Sarah's going to be mad at me if I don't know this, but and my understanding with Ogatia polymorpha was that the mating type loci are really close to one another. So you've got the A and the alpha gene really close on chromosome 3, and those can switch in and out. So it would be interesting. It's interesting to me that they're on completely different chromosomes, and how that could work, and how that organization. Sarah's turning on her camera now.
I can jump in here and talk about a little more. Yeah. So in this species Sturmera querculum, we think that it's possible that they're on the same chromosome, but the repeat region seems to be, at least in the short read assembly, a lot longer potentially. And so we're trying to figure out what the actual situation is with that species.
Okay.
So it's possible they're on the same, they're definitely on separate contexts, but it's possible they are on the same chromosome. We just need a better assembly to actually verify that's what's happening.
I thought it said chromosome there, chromosome A and B.
I think it was labeled chromosome A and B, but I guess we don't know if they're chromosomes or contigs.
Okay, got it. So they
could be because the short read assembly is a bit of a mess for it, because it seems like it's a little bit more complex even than some of the other species we've worked with.
Okay, that's fine. I was just curious because I didn't know how far along you had gotten to form the genome yet, or if you established that as much, but. Okay, that's good to know.
That's why we really want to work on that one with long reads, so that we can try to figure out what the actual structure is, because it's been a little bit difficult to figure out from the short read assembly alone.
And it sounds like you just got the long read data like a week or two ago. So
yeah, we were just in the very early stages of getting things put together, so the assembly is not finished yet. But yeah, we're gonna dig into it hopefully over the summer.
That sounds fun. That sounds fun, because I'm guessing that the OHAG, and also the OHAG one too, is going to be very interesting to see, the long read ones, because I know Tiffany presented earlier, but we actually got some of the contigs put together in a pretty good-ish organization. She was able to compare some of those. So
great, yeah, we got long read data for that too, and we're also working on the assembly for that as well. So it's all in the works.
Lots to do.
Yep.
Awesome. Hey, it's 3:21. Let's go ahead and call it a day. So. Thank you to all the presenters in session 2, room 2. Thank you very much for your time, for dedicating a Saturday to come share your work with us. I think it's great that all of these universities are collaborating together on this regional seminar series, and it's great to showcase the research that's ongoing. So thank you, everyone, and have a great weekend. And I guess this concludes session 2, room 2. Thank you again to all the speakers, and have a great rest of your weekend, everyone. Thank you, everyone, for your for your time to chat. So thank you very much, everyone, and we'll see everyone hopefully on campus or in person in the near future. So thank you much, and have a great weekend, everyone. Thank you.